We're on the component 1 Taskbook, Energy for Life, and we're on to our photosynthesis section. Question 3. Well, this is a hilarious question, quite tricky. If you can get kind of over half marks in this, then you've survived quite well. It starts off okay, and this is probably where you get most of your marks out of this 10. And place a tick. So it doesn't want any crosses, it just wants ticks. And it's the difference between the light and the dependent and the light independent. And you just need to think about my light dependent reaction, splitting of water, producing oxygen, ATP, and reduced NADP. And it occurs on the thylakoids in the chloroplast. So that you should know this stuff. So in the light dependent reaction, yes, that's producing oxygen, it's producing ATP, okay, and it's producing NADPH or reduced NA, so that's a different word, that's using rather than producing. It's on the thylakoids, so not in the stroma. Okay. Presumably on the other side, this will tally up. If it doesn't, we need to go back and have a think. So where we're fixing or using carbon dioxide, incorporating carbon dioxide, that's the dark reactions or light independent. And just double checking these, we're not making oxygen in the Calvin cycle. We're not producing oxygen in ATP. Yeah. Yeah, reducing, using the reduced NADP or NADPH, using that in a Calvin cycle, yeah. And in the stroma, yes, so that's in the stroma there. Right. Before we go any further, let's just have a look at the Calvin cycle. Let's start off with our 5 carbon sugar. 1, 2, 3, 4, 5, and we've got a 1 carbon there. We have a 3 carbon there, a 3 carbon there. Okay. So Calvin cycle. This isn't balanced. You can find balanced ones on the internet, and you can find ones with all of the various steps in. There's lots more steps in, but this is a basic thing that we know. So we're getting this 5 carbon compound, and we're adding our 1 to it. So, and this initially produces a 6 carbon. 1, 2, 4, 5. Yeah, I'm going to put it there. A 6 carbon intermediate. And then this almost immediately separates into 2 3s. And then we've got that 3 there, and then that's been regenerated. And some is coming off here. On average 1, but it doesn't, if you do the balanced equation, it works out. And this is where our glucose and so on. Okay, so we've got this is a 5 carbon, this is a 1 carbon, this is a 6 carbon, this is a 3 carbon, and this is a 3 carbon, and then we've got all sorts of stuff, glucose, starch, and so on at the bottom. Names for these, well, we've got our 1 carbon is CO2, our 5 carbon is ribulose bisphosphate, or RUBP. This is a step that you might not know much about, but we know this in our textbooks. This is called GP. and this is called TP. So triose phosphate. So I refer you to Miss Knight's excellent video on the Calvin cycle. Other books and other diagrams might give slightly different names to this. TP, that's sometimes called GALP or glyceraldehyde 3-phosphate. Glyceral, I can't spell it, glyceraldehyde 3-phosphate. I can't even bother to write it there. And this is sometimes called glycerate phosphate. Glycerate phosphate. So you can see where some of the confusion might arise with some of the names. I think with your exam questions, they'll give you the ones in the style that they've given you as part of the spec. And these are the terms that they use in the spec. We've got 6 carbon here, and 6 carbon things are generally called hex. So hexose something or other. Okay, and that's where we are at the moment. And what Calvin did was Calvin had, I don't know where to draw this diagram, I'm going to draw it here, had a lollipop apparatus. So there's a lollipop apparatus, and this was full of algae, chlorella, single-celled. Think of single plant cells and you're not too far off. Single plant cells. And shone a light on that. And they photosynthesize quite happily. You're using carbon dioxide, producing these, and this is spinning round and producing glucose. Every now and again we can squirt in some radioactive CO2. So we can squirt in some radioactive CO2, and this is just like a little flag. So it's just got 2 extra neutrons in the nucleus, but it's radioactive. So when we put it next to photographic paper, it makes it go black. A bit like X-rays and things like that. So it just makes it go black. And then the only other modification to this was that there's a tap on the bottom, and that goes into some evil substance. Hot solvent, ethanol and methanol, that will instantly denature enzymes, stop any metabolic processes, kill whichever organism is unlucky enough to get squirted into it. But this does allow you to squirt in some radioactive CO2, let it do whatever it's going to do for however long you want, and then stop the reaction. Then what happened was they take this solution and then do what you did. They make a chromatogram with an origin, they put the blobs on there and then see what blobs come up. They actually did this 3D using 2 different ways, but we won't go into that now. But they do chromatography a bit like what you've done in class. And so we get to this. If you can imagine the situation where we've got some radioactive CO2, I'm just going to make this radioactive, then what's the first thing, if we get that radioactive carbon there, what's the first thing that's going to happen? It gets incorporated into our hexose. Okay, so that's the first thing that's going to happen to it. Here's a chromatogram, and the blobs on this are pretty hard to see. We've got a blob there. I'm just going to circle these blobs. There we go. And so if we stop this reaction pretty fast, we just tend to get this one blob. Okay. Why are the blobs going different distances up? Well, it's the same reason you know from school or in your gel electrophoresis answers. Smaller compounds will go further. So these are C5, C6. You know, these are twice as big as these other ones there. So it's a bit of a red herring to talk about how far they've gone. It's just a method of separating them. What we're more interested in is this time scale here, and which ones have we got. So are they present or not? So this time scale and whether they're present or not. And so a short period of time, what have we got? We've got the bigger ones. And so we've got some of this hexose here. I think it's a bit of a red herring to think about IUBP at this stage. But certainly you can imagine that being incorporated into this 6 carbon, which then pretty quickly, 5 to 30 seconds, we get a second blob appearing. So that's 1 to 5 seconds. 1 to 5 seconds, and then after kind of 5 to 30 seconds, our hexose is quickly broken down, and we've got there's our radioactive CO2. It's now in GP. And there it is. And then after 1 to 5 minutes, we're seeing this other stuff appearing, so it's being passed on. There's our radioactive carbon. And this blob at the bottom is getting dark. Well, this is now spinning round, and it's got this far now, and we're making more and more and more of that, and then presumably in our glucose will then appear, and in our starch will also appear. And by that point, all of the blobs for everything will be dark. Okay, but we can see this over time. And now we need to try and answer the question based on that rambling discussion. Suggest why the suspension was run into hot ethanol. The simplest sort of thing to think about is it kills the algae, but we need a little bit more than that in this. What actually happens is it stops the reactions of photosynthesis and denatures enzymes, particularly kind of ribisco, that sort of thing. So stops photosynthesis reactions, denatures enzymes, that sort of thing. Essentially kills this algae and stops whatever's going on. Suggests one biologically important similarity between the two forms of carbon dioxide. The important word here is biologically. And so it's trying to hint in bold, it's trying to hint, please don't tell us the chemistry of this. Please don't tell us that it's got 2 extra neutrons. Save it for your chemistry exam on isotopes. Biologically, we're using a marked version of something, and we're hoping that our marked version also does exactly the same as our original version. That's what we're wanting. So we're wanting, photosynthesis occurs with both types equally, or photosynthesis, it doesn't matter whether it's radioactive or not. So both can be fixed identically in photosynthesis. So essentially we want this algae not to care whether it's using radioactive CO2 or normal CO2, if algae cared. And with reference to the diagram, 3 conclusions that can be drawn from the appearance of the sequence. Okay, so not how far, but the appearance. So it wants to go, tell us about only one blob here, 2 blobs, 3 blobs. That's what it wants. So pretty tough. So where does it first appear? It first appears in these larger compounds here. So first appears in hexose. And so it kind of suggests that it combines with the 5 carbon to produce this 6 carbon. So it suggests that CO2 combines with RuBP to form a hexose. So, the 1 carbon plus the 5 carbon goes to the 6 carbon. So that's the first thing. And then the next step, we're trying to explain this next thing. So then we did have a 6, but now we've got a 6 and a 3. So it implies it splits next. So the 6 carbon, the hexose splits. And from your knowledge, that would be to produce GP. And then the next, you say, well, then this forms another 3 carbon. So then, forms another 3 carbon compound. And from your knowledge, this will be triose phosphate. So a bit tricky that. A bit tricky. Even if you knew about this, there's different words for the compounds and a little step which you might not have memorised. So pretty tough that. You'd certainly do these 2 up there. Well, certainly the first one, and you'd have nailed these 5 there. So you'd have got a kind of C grade from this, even without this nonsense down here.